cancer cases. As we know that cancer is a progressive disease that usually begins with the increased frequency of cell division. So all cancer result from changes in the DNA sequences of our genome. These changes occur throughout life because the genome within our cells is exposed to some sort of mutagens like UV radiation, and these will accumulate the mistakes or the, let's say, the deficiency during the replication. These changes result in a progressive, subtle divergence of the DNA sequences from the original copy from the fertilized egg. So one of these mutations alters the function of a critical gene, and that will provide a growth advantage to the cell in which it has been occurred. And this means that this cell and its offspring divide at a faster rate than that of their neighbors. The result is tumor formation, invasion of the surrounding tissue, and eventually the metastasis or the spread of the cancer to the other parts of the body. So as you can see here in this tissue, that some of the places is indicating the presence of the tumor. And when you want to see the cancerous cell under the microscope, you will detect it as increased cellular and nuclear size. The cells will be the shape of the cells will be abnormal, the size will be abnormal, the even the proportion of the cells that goes mitosis will be also increased. So typically they will lose their normal shape and the tissue organization. So uh, it will just uh, have what's called dysplasia, where the tissues will be increasing their cell division and they will be abnormal in organization. And eventually, as we said, these cells will exhibit what's called a metastasis, where it will expand to the adjacent tissue. For example, here, these normal cells are organized in the normal shape. Then it will have some dysplasia, where the organization will be differing. Then it will go severe dysplasia, where you can see comparing to the normal is totally different. And finally, once it's completely became a tumor, it will just spread to the other tissue in what's called a metastasis. If you will just see or notice this karyotype, the normal karyotype will eventually have 46 chromosomes. While here, as you can see, it will have, if you count it, it will be 78 chromosomes. Some chromosomes will be presented in not only two copies. For example, here, chromosome 1, we should exhibit only two chromosomes with a yellow color. Here we have 6 chromosomes with variable colors. It means that there are some mutations or some uncontrolled division, or let's say some of the let's say what's called interchromosomal rearrangement, where the chunks of these genomes has been broken and then joined to other parts of the genome, will be presented. And this how you can see or visualize the karyotype of a cancerous disease. For example, here this is a karyotype from a tumor tissue from a breast cancer. So what are the carcinogen? Carcinogen is any agent that will cause the cancer or will increase the possibility of having a cancer. Uh, Not all the carcinogens are mutagens because we have uh, defined the mutagens before, but not all of them will cause mutations, but mostly they will increase or will have the high incidence in having a cancer. Uh, some of them, they don't directly damage the DNA. They will have some substances that will just make the division of the cell more rapid and more fast. and it will leave the less opportunity of the cell to repair itself or to correct the errors in the replication. Therefore, this division will go with no control, having these defect or damaged DNA without any correction. Uh, they might be biological, physical, or chemical in nature, and any gene leads to an uncontrolled increase in cell division. We can call it oncogene. For example, here, if we have a normal cell and we have some carcinogen or some cancer-causing agents that are exposed to the normal cells, you will have in your DNA what's called proto-oncogenes. These are the genes that can develop a cancer once it's activated. So once it has the agent or the let's say the oncogenes, it will be activated as oncogenes, and therefore they will develop into cancerous cell.
Cancer is a non-infectious disease. It was the #1 killer in Hong Kong. How does cancer develop? Cell division is normally well controlled by certain segments of DNA in cells. However, when mutations occur in these segments, cell division may become overstimulated or never be inhibited. This produces a mass of abnormal cells called a tumor. When further mutations occur in a tumor cell, the tumor cell has an increasing tendency to divide and becomes a cancer cell. A malignant tumor forms. It destroys nearby tissues and develops its own blood vessels. Some cancer cells may enter the bloodstream and spread to other parts of the body. At the new site, the cancer cells divide to form a new malignant tumor.
So what are the carcinogens? It could be biological, as we said. The biological factors can be linked with cancer, for example, human papillomavirus, the HPV. The HPV is a virus that is recognized as a cause of cervical cancer. The HPV mainly is a sexually transmitted virus, which has more than 100 different strains. 30 of these can cause cervical cancer and genital warts. So when HPV enters the cell of the cervix, proteins made by this virus activate and inactivate the oncogenes and tumor suppressor genes respectively. And it is like a machine for turning on lots of cancer genes. So these viruses will just trigger these oncogenes and will stop or inactivate some tumor suppressor genes. And as you can see here, the picture, this is the HPV virus, and here these are the normal cells, while here you can see that these are the cancerous cells that were developed out because of this HPV virus. And there were more than 5,050 new cases of cervical cancer and approximately 260,000 deaths from cervical cancer worldwide. So it's considered the high frequency cause of death if we are talking about the cancerous cases. The majority of these were from women developing in the developing country where cervical cancer screening programs are often unavailable and healthcare infrastructure was weak. So some of the countries, the presence of this HPV cervical cancer has led to the development of some vaccines against the HPV. So the vaccination will reduce the cases of cervical cancer, and it is regarded by cancer experts as a stunning advance in the prevention of the cancer. So as you can see here that the HPV will develop some or will be the reason of some cervical cancer in a human, or especially the woman. Other causes or carcinogens will be a physical one, which are the radiation. The first known or the most frequent radiation source that will cause the cancer are the ionizing. So they will induce some damage to the DNA within the body, and these will just skip the, let's say, the correction, the error correction, and will have these damaged DNA molecules that will be cancerous, and then they will they will develop into tumors. Another type are the chemical carcinogens. Uh, They either can be natural or synthetic. These are some of the classes of the carcinogens and some of the examples. For example, here the azodides group, some dyes and pigments used in the textile, leather, and paints might cause or might be a chemical carcinogens, or might cause some cancerous cell, some inorganic compound that will induce some chronic inflammation and reactive oxygen species like the asbestos. These are mostly the most causing chemical carcinogens. So, as we said, what are the characteristics of the cancerous cell? They are resistant to apoptosis. Now, apoptosis is the cell death. As we know that when there is a damage in the cell and or damage in any part or any, let's say, process, it will go or it will be sent to apoptosis to get rid of the damaged cell. Therefore, these cancerous cells will not have any apoptosis, and they will resist the apoptosis. Therefore, the damage will not be corrected, or you will not get rid of this damage from the cell. And they have an uncontrolled ability to divide. They are immortal, they never die, and they divide in an increased rate. So they have a high frequency of division rate. They are insensitive to anti-growth factors. So once you want to, let's say, treat the cancerous cell or the tumor with anti-growth factors, like to suppress the division, it will be not sensitive to these kind of factors or treatments. So this is another characteristics of the cancerous cell, and it can invade to the neighboring tissue in what's called the metastasis, and it will go secreted. It will have a secretion that will digest the extracellular matrix material, and they will invade to the other tissues. And finally, they can secrete some chemical signals that simulate the growth of new blood vessels in what's called angiogenesis. What is the tumor genesis? It's the genetic changes that is associated with the tumor genesis can be divided into two categories. It's either we consider it as a gain of function or loss of function. The gain of function will involve the activation of some oncogenes. As we said, you have to get some proto-oncogenes to be activated and converted to oncogenes. They will lead to let's say, have to give the cell a high rate of growth and division, so they will be some sort of hyperactive. The other category is loss of function, where they involve the inactivation of tumor suppressor gene. These genes will inhibit the cell growth, and which cause cancer when they are not expressed. So it might be a result of gain of function or loss of function. Uh, there are some non-lethal genetic damage that lies at the center of carcinogenesis. It could be loss or damage to suppressor genes, as we said. It could be overexpression of some promoter genes. The promoter genes, as we know, these are the genes that gives the start or the ignition for the some processes. So as long as you have overexpression of these genes, you will have more division rate. Sometimes it is loss or damage of apoptosis genes, so the cell will not go for apoptosis uh, stage. So the damage will still presented there, and sometimes it will be a loss or damage of DNA repair genes, where you have any error in the DNA. It should go for some sort of repair, but because of the cancer, you will not have this ability to repair the gene or the error in the DNA. So all of these will have some causes or some reasons to cause the cancer. There are mainly two types of the cancer genes. It could be a tumor suppressor genes, as you said, and it could be oncogenes, sorry. Uh, we know that we might have 400 somatic cancer genes, sorry. Uh, as we know, the somatic cancer genes are the cancers that happens to the somatic cells, that they are available in whole the body except for the egg and the sperm. If you want to, let's say, check for any somatic mutations, some of the genes behind some type of cancers, you just go to the cosmic. It's a catalog for these type of mutations that are causing the cancers. Just click on this link and it will take you to the catalog, where you just define the type of the place of the tissue, the type of the cancer, and it will give you all the information you need about that cancer, the genes involved, the frequency of having this gene involved in this cancer. It might be, let's say, 5% of the cases that they have a defect in the certain genes related to this cancer, and so on. If you need a check for the information there. Oncogene will code for proteins that drive cell division, which then these genes will acquire mutations that result in continually active proteins. They become oncogenes and cause the uncontrolled cell growth and division. So these oncogenes will be responsible for the controlled process of the cell division. So, for example, if you have a mutation in one of the oncogene, this will cause a cancer. So one mutation, one copy will be enough to have a cancer based on the mutations in the oncogene or the proto-oncogene. As we said, that the activation of the oncogene, we have proto-oncogenes, or which called pre-oncogenes. They will gain of function of mutation, so they will have a gain of function, and they will be developed to oncogenes or cancerous cells. If there are proto-oncogenes and they didn't have any mutation or any carcinogens, then they will be normally regulating the cell growth, and they will be considered as normal. So as long as they don't face any type of mutation or let's say targets, then they will not develop into cancer. Thus, is an example for the some of the proto-oncogenes. Normally, the RAS in the inactive form, prior to the simulation of the pathway, the components will include some soluble ligands, EGF, its receptor, the EGF receptor, a, a tyrosine kinase, a RAS, a G protein, for example, several kinases like the RAF, the MEK, the MAPK, and a transduction or transcription factor, which is the TF. Here, for example, the active pathway, as it's shown, will be binding of the ligand to its receptor, lead to the auto-phosphorylation of the receptor, and therefore, through series of proteins, the phosphorylated stimulates the conversion of RAS into its active GTP bound form. So this activated RAS then simulates the phosphorylation of series of kinases, which ultimately activates the transcription factor and the expression of the gene requires for the cell proliferation or division. So once there is, let's say, a mutation in this RAS, it will be not it will cause it to be in the permanently active form. So it will just lead to the uncontrolled division of the cell. Nothing will tell the RAS to stop, and it will be always active. So this will lead to the constitutive activation of the cell cycle and cell division. The other thing or the other type of cancer genes are the tumor suppressor genes, which code for proteins that uh, slow down the cell growth. They can halt the cell growth cycle to stop unnecessarily division or promote apoptosis in the cell if the cell DNA is damaged. There are different tumor suppressor proteins that carry out the following functions like repression of genes which are essential to the cell cycle, so they will inhibit the cell division. They are linking the cell cycle to the DNA damage. If there is a damage to the cell, it will not allow it to divide, for example, identifying where the DNA damage is, and it will initiate the apoptosis or the cell death. Uh, so it will prevent the cell growth or cell division. It's not like the oncogene, as you know that the tumor suppressor genes, they have to be mutated in both copies, so they will cause the cancer. It's not like oncogene that one mutation in one copy will be enough to cause the cancer. We have more than 30 genes that are classified as tumor suppressors, and as we said, their normal function is to send the cell for a programmed cell death, or they will repair the DNA, as we said, and that will prevent the abnormal cell division. If there is a loss of this function, then this will cause the cancer. So one example of the tumor suppressor genes is the BRCA1, and the breast cancer gene, which is responsible in the DNA repair. So once there is any mutation in this gene, it will just cause the cancer. Another important tumor suppressor gene is the transcription factor, which is p53. So other proteins in the cell will sense the DNA damage or abnormalities in the cell cycle, and it will activate the p53 through several mechanisms, including the phosphorylation, as we said before, and transport into nucleus. and an active form, the p53, will induce the transcription of genes with several different types of tumor suppressing functions. They will repair the DNA, they will arrest the cell cycle to a certain stage, and they might send the cell for apoptosis. Uh, Most of the tumors, like 50% of the tumors, they will contain some sort of mutation in the p53 tumor suppressor gene. So people who inherit only one function copy of the p53 have a greatly increased incidence of early onset cancer. As we said, the p53, you need two mutated copies of the chromosomes to have a cancer cell. So once you have one mutation, and it will be inherited, so there is a high possibility of developing a cancer case. However, as with the other cancer related genes we have discussed before, most mutations in the p53 are sporadic rather than inherited. So they will be only in the somatic cell and it will not be inherited. So mutations of the p53 through formation of pyrimidine dimers in the gene following exposure to UV light, for example, has been causally linked to the squamous cells and basal cells carcinomas, for example. So here we have normal growth for the cell. It will have p53 as protector, so it will arrest the cell growth and it will send it uh, for DNA repair, and then it will cause the cell survival, and it will has the anabolism. Once there is an oncogene growth, the p53 will be having the loss of the function and it will eliminate the cell without any without any correction, and then it will be a tumor, for example. So again, in the normal p53, the DNA damage, uh, DNA cell cycle abnormality, hypoxia, they will all have the normal p53, which, as we said, it will arrest the cell cycle, then it will send the DNA for repair, and again the cell cycle will restart. Otherwise, it will be sent for apoptosis or cell death. So once there is any damage in the p53, the cell cycle will continue and the cell can become cancerous without any correction. So these are some of the cases of the abnormalities that the cell might face, and it needs the p53 for a correction. So once it is not corrected, then the cell will differentiate or will divide with this type of error without any correction. Concept of these genes related to the cancer, we can take the car as a model to understand the concept. For example, we can consider the tumor suppressor genes as the brakes for the car. The car has two brakes, so once you have one brake damaged, the car still can go forward and can do its function. Once you have two copies, it means that you don't have a brake for the car, so the car will just go forward with no stop like here. It will just go without stopping because there is no brake to hold the cars. If we want to take the concept of the proto-oncogenes, we can consider them as the gas for the car. So once you have high gas, or you will just press the pedals for the gas in the car, and there is a damage in it, there is only one pedal for the car for the gas. You can just press with your feet, and it will go forward. The same as the oncogenes, you will just have one mutation in the one copy of the chromosomes, and that's also will cause a cancer. Here also the same. Once you have one pedal or the gas has a defect, then the car will go non-stopping and it will just lead again to the cancer. So too much gas or too much oncogenes will lead to the uncontrolled division, and no brakes or no suppressor genes will lead also to cancerous cells. Changes that might occur for the cell too, so we can call it's a cancerous cell. The immortalization, where they will divide very fast and high frequency. The transformation, where they will just look for some source of blood supply, and through the process of angiogenesis, so they will have a growth of new blood vessels. Then the metastasis, where they, let's say, differentiate or travel to another tissue. So these are the major changes that you will see in the cancerous cell. So if you can see here, the mutation in multiple cancer genes are required for the development and progression of the single cancer. For example, you will start with the benign tumor, as you can see. The benign tumor, as you can notice, it will be in situ cancer, so it will be localized over here in one position. After that, the invasive cancer will appear, where it will look for blood vessel, and finally the metastasis cancer will happen, where it will travel to the other parts of the body or other tissue. So the multi-step model for the development, for example, of colorectal cancer. The colorectal cancer is the cancer that happens to the colon. So the clonal origin of tumor, each individual cancer is a clone that arises from a single cell. For example, here you have the colon wall. The normal colon epithelial cell will have some abnormal growth. Then the cancer will start to develop. First of all, the loss of tumor suppressor gene, the APC, will appear, that will cause to the growth of the benign tumor or the polyps. Then the activation of the RAS oncogene, here it is related to several, uh, let's say, several type of genes or several categories of genes that will cause this type of uh, cancer. First, it will be loss of the tumor suppressor gene APC, then the activation of the RAS oncogene, where this polyp will just develop to adenoma, and then again it will have a loss of tumor suppressor gene, the DCC, to develop to adenoma, and again they will have a loss of tumor suppressor P53 that will cause the malignancy. External causes of cancer can be natural radiation, for example, such as UV rays from the sun, which is known to be the primary cause of skin cancer, for example. Through prolonged unprotected exposure to sunlight and UV light in the form of sunburns, for example, we increase our risk of developing skin cancer. And that's why you see the lifestyle of the newly, let's say, youth people, they look for these tanning or these sunbeds, so that might increase the developing of skin cancer. The malignant melanomas are the least common type of skin cancer, but the most serious ones. So there are around 10,000 new cases each year in the UK, around 60,000 in Europe, and around 54,000 in the USA, as the statistics says. So malignant melanoma is responsible for 3 out of 4 skin cancer deaths. Most forms of skin cancer, however, are relatively treatable, especially if detected early. So these exposure to the UV light will have some sort of UV light induced mutation, leave a typical signature of C to T or G to A mutations. They are point mutations. These form the vast majority of the mutations. So these specific changes occur as the DNA damage from the UV radiation leads to the formation of covalent bonds between the two agents, pyrimidine, the C and the T, and the DNA molecules. So these will not be corrected, and therefore they will develop to the malignant carcinoma. There are other external causes of cancer, like, for example, the smoking, where the chemicals in the cigarette smoke have been causally linked to the development of lung cancer. Lung cancer causes around 1 million deaths worldwide every year, almost all of which are associated with smoking. Tobacco smoke contains more than 60 mutagens, for example, that binds and chemically modify the DNA and mutate it. These brand the lung cancer genome with characteristic mutational pattern. For example, some of the researchers have shown that the tobacco smoke carcinogens, polycyclic aromatic hydrocarbons, which are the PAH, for example, and the nicotine-derived nitrosamines cause the G to T point mutation. So the G will be converted to the T in the smoking mutations. Uh, Another type is the diet of the person. They might cause cancer if the person is overeating, where overeating will lead to obesity. The obesity is known to be associated with bowel and stomach cancers. If the obesity can be reduced, then the number of cancer cases will also be reduced. Uh, So here you can see that there are some cellular events that occur to activate and deactivate the KRAS, for example, which helps to transmit the external growth signal to the cell nucleus. So it will cause the normal cell growth. So when uh, it will be activated when it binds to the GTP, and the inactivated or switched off when it's bound to the GTP is hydrolyzed to some GTP form. So the KRAS is a guanine nucleotide binding protein that acts as a self-inactivating signaling molecules. So the normal, as we said, the KRAS normal can be activated by signals from cell surface receptor that promote the exchange of bound GTP to GTP. The KRAS is switched off when the GTP is hydrolyzed. So the KRAS oncogene has mutation that result in amino acid substitution in the key region. So these mutated genes will encode to some proteins with reduced GTPase activity. These will cause, along with other mutations, can lead to the development of cancer. So as you can see here, some exchange factors will happen, and this will be hydrolyzing the GTP to the GTP, and therefore will cause the cancer. These are some of the classes of oncogenes and tumor suppressor genes, for example, the classes, some of the examples, and some of the incidents that they might cause. For example, here, the most known one these days is the breast and ovarian cancer with the BRCA1 and 2 and P53. Class is the DNA repair and cell cycle checkpoints. These are considered as checkpoints, so if there is any mutation within them, then the cell will just have some problems, and that might cause cancer. Another example uh, of these cancer that might be caused with one of the mutation is the chronic myelogenous leukemia, or the CML. These these happens in some sort of translocation between the chromosome 9 and chromosome 22. So these translocation will involve the breaks in two genes, the CABL and the BCR, as we explained before. So the fusion or the translocation break result in the chimeric genes, which is called the BCR-ABL, or the Philadelphia chromosome, that contains exon 1 and 2 from the BCR, and 2 and 11 from the ABL, and it produces chimeric protein that is transcript like the PCR and contains the ABL enzyme sequences. So this chimeric protein has a tyrosine kinase from the ABL gene sequences that is unique to the CML mutant cell.